We'll be going over proto-oncogenes and tumor suppressor gene, and then we'll be focusing on epigenetics and how epigenetics influence cancer. So in this image here, I'm giving you a normal cell. This is a fibroblast, and we can see that there are growth factors. These are growth factors. When they bind to this cell surface membrane receptor, it causes this this fibroblast cell to undergo cell division, i.e. proliferation. So proliferation means cell division, they're dividing. Essentially what's happening is you have proto-oncogenes. These are genes obviously found within the nucleus. So this is the genotype. Proto-oncogenes, they code, they make growth factor. So proto-oncogene, they make growth factor. So these growth factors here are made by which gene, are coded by which gene? Proto-oncogene. And these growth factors will bind to the receptor of cell and is going to activate the cell to start dividing. So how does this relate to cancer? So essentially what's happening is, in cancer, this process, these growth factors are always binding to the receptor, i.e. the proto-oncogene is continuously, always, all the time, coding for growth factors, which is causing uncontrolled cell division. So this proto-oncogene, when it mutates, so mutation causes a proto-oncogene to be always stimulated, to be always switched on, and we call this oncogene. When that mutation has occurred and the proto-oncogene is doing too much, uncontrollably too much, non-stop, making lots of growth factors, causing increased cell division, we call this oncogene. The example I use is a car. So you have the accelerator. Accelerator is always turned on, is switched on permanently, so the car keeps moving non-stop, just like your cell non-stop continues to divide. So proto-oncogene, just a recap here, these genes are turned on, or they are always activated. And when they become activated, turned on permanently, is called a oncogene. And this leads to uncontrolled cell division. So proto-oncogenes, all of us have it. So even if you don't suffer from cancer, every single person has proto-oncogene. Because when you want to grow your cells, when you want to repair your cells, you will need to have proto-oncogene making growth factors or coding for growth factors, and this will cause increased cell division. But we want it in a controlled fashion. We don't want it all the time, only when it's needed. With cancer, it's all the time. Hence, oncogene causing uncontrolled cell division. RAS is an example of a proto-oncogene that can be mutated into a oncogene causing increased cell division. Let's do a quick exam question. Have a read and pause the video and see if you can answer this question. So KRAS gene codes for a protein called KRAS. This protein relays signals from outside a cell to the nucleus, stimulating cell division. Alteration of KRAS gene produces a oncogene, which can cause a tumor to develop. Suggest and explain how alteration of KRAS gene can cause a tumor to develop. It's a 3-mark question. So you need to talk about mutation. So KRAS gene has mutated, and after it has mutated, it has changed the function, meaning there's more signaling. Remember, the KRAS proto-oncogene, when it comes to oncogene, it is not switched off. Rather, it's the opposite. It's switched on permanently. So whatever you was doing before is doing the same thing, but more. And that results in rapid or uncontrolled cell division, hence causing a tumor to form, a mass of cell. Tumor suppressor gene is another gene that is important and can cause cancer if it's mutated. Tumor suppressor gene, essentially what they do is that they stop cell division or they slow down cell division. So we know that our cells are not always dividing. They only divide when it's important or when it's needed. So these tumor suppressor gene, they are the gene that is slowing things down, causing the cell not to divide unless it's needed. If it's not needed, the tumor suppressor gene says don't divide. And also, if you have repairs in the gene and the DNA, the tumor suppressor gene fixes those repairs. So let's say you have a mutation or you have other stuff going on, or an injury to the DNA, the tumor suppressor gene will try to repair those problems. If the damage is too much, okay, then the causes the cell to die. It's called cell programmed death, apoptosis. So in cancer patient, this tumor suppressor gene is switched off, is turned off permanently. And this means what? That means remember that the normal function was to stop cell division, to slow down cell division. So this causes uncontrolled cell division. You can think of this as an example of a brake in a car. So the brake is not working. So you have uncontrolled cell division. Examples of the tumor suppressor gene are TP53. This is a tumor suppressor gene that is mutated in about 50% of all cancers. In breast cancer, you have BRCA1 and BRCA2, which are mutated in them. So these are three tumor suppressor genes that I have shown you here. Let's go into epigenetics. Have a pause and what do you think? What do you feel about these four options? Can our environment and our lifestyle influence the following four? Our aging, getting cancer, getting disease, and can we transfer genetically this to our offsprings? The answer to all of them is actually yes. So epigenetics. I'm going over here your transcription, because you need to know this quite well to understand this module. We know that we have a transcription factor in the cytoplasm. That's our molecule. This molecule, it leaves the cytoplasm and it enters the nucleus. Okay, and we know that it can be an activator or repressor. In our example, in epigenetics, we'll be focusing only on the activators. The transcription factor will find a gene, find a DNA, and that DNA has to be accessible. So therefore, the transcriptional factor can bind to the promoter region, and that will cause the RNA polymerase to bind to the promoter region and initiating transcription. So the key message here is the transcriptional factor must bind to the promoter region, which means the DNA has to be accessible. The transcriptional factor must have access to the DNA. If the DNA is hidden away, is wrapped around tightly, then you will not have the gene being expressed. Reason because the transcriptional factor cannot bind to the promoter region, cannot bind to the DNA. So it will not initiate transcription. That's the main important point for epigenetics, is the accessibility of the transcription factor to the DNA. I'm showing you here chromatin. This is our DNA, that line here is our DNA, and DNA is actually wrapped around a protein called histone protein. Histone is charged positively, DNA is charged negatively, and well done, yes, positive, negative cause attraction. So therefore, DNA wraps around histone very nicely. And that is called chromatin. So DNA histone complex is chromatin. Now, I have two boxes here. First box, if the DNA is wrapped around really, really tightly, we call this more condensed, then the transcriptional factor cannot have access to the promoter region. The transcriptional factor cannot bind to the DNA. So that will cause the DNA to be switched off. So that DNA or that particular gene will not be transcribed. In the other hand here, we have a DNA histone complex that is loose. So it's less condensed. If it's less condensed and is loose, that means the transcriptional factor does or can have access to the gene, which will cause it to bind to it, which will encourage RNA polymerase to bind to it, hence initiating transcription, so the gene is switched on, i.e. the gene will be expressed. Two important words which I didn't put down. If the chromatin is really very tight and condensed, it's called eochromatin. If it's loose and less condensed, it's called heterochromatin. And that you need to know for your AQA A-level exam. Eochromatin and heterochromatin. In epigenetics, we'll be talking about how our chromatin can become very tight and very loose. This is our histone, and this is our DNA wrapped around it. On this histone, or on the DNA, we have chemical tags which are covering this complex. So chemicals can be attached to our DNA histone complex, and this is called epigenome. Meaning epi means on, so chemicals are attached to the DNA histone complex. Epigenetics is a study of inheritable changes in the gene function that have not been caused by changes of the base sequence of DNA. You have to memorize this definition for a 2-mark question for AQA A-level epigenetics definition. So let's see how it occurs. I'm showing you here acetyl group. Acetyl group is negatively charged, and it binds to your histone. So these orange shaped pentose shapes, they are showing you that a acetyl, this orange is acetyl is added to the histone. That's called acetylation. So acetylation is the adding of acetyl group to histone proteins. If you have increased acetylation, what does that mean? Lots of acetyl added to it. This makes the histone less positive, which means that you have a complex that is that is less condensed because the histone is less positive. For the A-level exam, you don't need to mention about the less positive, more positive. I put it here just to make sense slightly bit more. So when you add lots of acetyl, the DNA histone complex is less condensed. That means it's very, very loose. If it's very loose, let's go here. That means it is accessible to transcriptional factors, which means your transcriptional factor can bind to the promoter region of the gene, which will cause RNA polymerase to bind to the promoter region, which in other words, you are transcribing that gene. So the gene expression is switched on. So gene is switched on. The opposite happens if you were to remove this acetyl group from the histone. That is called decreased acetylation. And the reason why opposite occurs, it makes the DNA histone complex here, so mistake, more condensed. What does more condensed mean? More tight. If it's more tight, transcriptional factors cannot bind to it. They don't have access to the DNA. So which means RNA polymerase cannot bind to the promoter region. So therefore, gene expression is switched off. Methylation. Methyl, this is a methyl group here. Now methyl this time, it does not bind to the histone protein. It binds to the DNA. And that you need to know for the exam. So methyl group here, as you can see, methyl group are added on to the DNA. In particular, they are added to the cytosine base. But most importantly, you should know, it binds to the DNA, not the histone. Increased methylation. So by adding the methyl group to the DNA is called methylation. Increased methylation, lots of them are added to the DNA. It makes the complex more condensed, very, very tight. If it's tight, transcriptional factors don't have access to the DNA, so therefore the gene is not expressed, switched off. Opposite happens if it's decreased methylation. If you remove those methyl groups from the DNA, it makes the complex less condensed, meaning less loose, which means transcriptional factors have access to those genes and it will be stimulated. It's going to be expressed. So that's decreased methylation. In the next video, I'll be going over exam questions relating to tumor suppressor gene, proto-oncogene to oncogene, and epigenetics.